So today we're actually happy to have Dr. Jenna Hershberger joining us. She's an assistant professor working on vegetable breeding and genetics at Clemson University. So it's a timely discussion for her presentation today. Coming off some of the questions that we were seeing in the group coming up from the last discussion. So Dr. Hershberger in the past was also a postdoctoral research associate, co-mentored by Dr. Miller at the Danforth Center and Dr. Schlottman at the Land Institute. And I guess that was her second chance kind of getting into looking on development and evaluation of new selection methods for applied plant breeding programs, including the use of spectral data sets to improve efficiency in the domestication of perennial grains and legume crops there. Prior, she had done her PhD at Cornell University in Dr. Gore's lab, where I think your experience started with this technology. So we're excited to hear more today about spectral data collection, analysis, and storage in plant breeding from Dr. Hershberger. It's all yours.
All right, thanks, Amanda. Okay, get this going. Okay, so Amanda just covered this, but I'm from Wisconsin. I did my bachelor's at University of Wisconsin. I worked with Phil Simon, USDA, and that's where I really got my start in plant breeding. And then I didn't start working with spectral data until my PhD with Mike Gore at Cornell, and then continued that at the Land Institute in the Danforth Center. So a little bit of review from what Amanda said, but okay, now I started about a year ago as a vegetable breeder at Clemson's PD Research and Education Center. So if you're coming to NAPB next week, you'll be up near the Clemson's main campus. We're actually about 3.5 hours away in Florence, South Carolina at the PD Research and Education Center. We've got a pretty big piece of land that we're working with. We've got lots of land for experiments and research. We are joined by a couple of people from the USDA, and it's a great place to be. So I hope at some point you'll be able to come out to visit us, even if not during the NAPB meeting next week. My program focuses on breeding for flavor, nutritional quality, and heat tolerance, but I am just getting started. So I did not inherit the program from anyone, and I'm starting from scratch working on butter beans and sweet corn. I won't be talking about that work today, but we'll focus more on broad topics related to spectroscopy. So most of you here are probably familiar with plant breeding. I think Trevor may have shown this slide even a couple of months ago during this seminar series, but plant breeding is a pretty cyclical thing. We go from selection, crossing, and evaluation throughout the plant's life cycles each year. And what takes up most of the time for most breeding programs is this evaluation stage where you're trying to decide in relative terms how well different individuals or families are doing so that you can make those informed selections to make more genetic gain. And a lot of the topics in the seminar series have been focused on how to speed up or make this evaluation phase more efficient. And spectroscopy is just another method that can be used to accomplish that goal. So one group of traits that are especially cumbersome to evaluate is quality traits. So we've got things like flavor, nutritional quality, texture, post-harvest storage. These things can require a lot of expensive equipment with a lot of training or reagents required. They can be subjective if you're using visual scoring. Sometimes they can require a lot of complicated logistics for keeping samples at the right temperature so they don't start to degrade, or you might be dealing with large things like cassava roots that you have to bring miles away to the lab to get analyzed. And all of this just results in things that slow down that evaluation process. And so the focus of my PhD projects were to evaluate and develop new methods to speed up this quality trait phenotyping process. And spectroscopy is just one way to do that. And then, oh yeah, I should have done that. So this is, an HPLC, one of the really highly accurate but slow and expensive methods used to do some of this phenotyping. So spectroscopy is a pretty broad term. I'm going to define what I mean by spectroscopy and how it's usually used in the context of plant breeding. So when I talk about spectroscopy, I'm primarily talking about visible and near-infrared spectroscopy. And so this is dealing with the region of the electromagnetic spectrum between 400 and 2500 nanometers. And when light from this region of the spectrum is shown onto a biological sample, the chemical and physical properties of the sample transform the pattern of light that's reflected back to the sensor. And this has to do with the different chemical bonds in the sample and them vibrating at different frequencies and causing differences in absorption of the light. But these signals, because we're dealing with many different bonds in the same sample, and these signals can be broad rather than like an individual nanometer. So the signals can start to overlap and they become pretty complex to try to parse out. So you end up receiving back from the sensor something like this figure on the right here, where we have our different levels of reflectance at different wavelengths throughout this region of the electromagnetic spectrum. And so we can use statistical techniques to try to parse out what exactly is happening and relate these spectral signatures back to the properties of the sample. And I'm going to talk a little bit about how we do that today. So a little background on spectroscopy. It's not a new technology. It was first discovered around 1800, but then has gained a lot of popularity more recently. It was applied agriculture by the USDA in 1968, and then throughout the 70s became especially popular within the forage community as a way to predict different forage quality characteristics. Over time, as spectrometers have improved the hardware and as computational capacity has improved, these have become more common and available and cheaper. So especially in the last few years, it seems that they've just become everywhere. So, but yeah, it's not new. It is an established technology. But as with any technology, there are some positives and also some limitations. So I guess reframing this from positives and negatives maybe as opportunities and limitations. So this technology can be fast and it doesn't necessarily require destroying samples. So it could be done on living plants or in a time series, because that same sample is can be kept. You can even reuse the same sample for different wet lab analyses after you've scanned it with a spectrometer. It doesn't require any reagents, and it can reduce the subjectivity, which is important when we're trying to phenotype things accurately for plant breeding use cases. But this technology has limited sensitivity. So if you're dealing with different compositional traits where you've got things at a really low level, like if you're measuring in just a few parts per million, it's probably not going to be able to pick it up. And this kind of has to be evaluated on a case-by-case basis depending on your trait. The method requires calibration, so you have to have some reference samples to be able to build a prediction model to use routinely. And this can also require pretty large initial investment if you're dealing with an expensive spectrometer or expensive method to generate those reference samples. and the data sets can be quite large. You generate a lot of data for each scan, and the files can become a little bit unruly and difficult to manage. So I'll talk a little bit about that today as well. So the process of using spectroscopy in plant breeding kind of goes through this workflow. This isn't entirely linear, but today I'll be talking about things in this order. So first we have to capture the spectra and then process them, get them all lined up, squared up, and then we can analyze them using many different methods. And then ultimately we need to be able to transfer and store these data types. So I'll touch on that as well. When we're thinking about spectral data capture, there are a lot of things that we need to consider before we start. So I'll talk a little bit about the spectrometer type, scale that you're capturing the data on, and then experimental setup. So first is wavelength range. So I mentioned that the visible and near infrared regions are typically defined as about 400 to 2500 nanometers. And within this range, different chemical components have different ranges at which they're detectable. So through decades of experimental procedures and lots of literature, people have kind of settled on these regions as the important ones for some of the different traits. So if you look down here towards the bottom of this blue square, we've got some regions that are important for prediction of or detection of chlorophyll, polyphenols, carotenes. And these make sense because, especially for carotenes, these are pigments. And so the area of the electromagnetic spectrum that has signal for these pigments is that yellow and orange region of the visible spectrum. Water has several different areas where it can typically be detected, nitrates, sugar, proteins. So this may not have you may not be able to look up your specific compound that you're interested in the literature, but I would definitely recommend trying and seeing if people have successfully been able to detect whatever you're looking for in this range before you start. And different spectrometers capture different regions of within this visible and near infrared area of the spectrum. So you'll want to make sure to note that when you're choosing your spectrometer. The next thing to consider is whether you need image-based or non-image-based data capture. So this figure kind of shows you've got image-based is you get that like X and Y spatial dimension for each spectrum of light. So you end up with this stack of images instead of like an RGB. Your typical image that your phone camera might take has 3 bands, R, G, and B, red, green, and blue. But then in a hyperspectral image, you end up with hundreds, even thousands of different wavelengths at each pixel. So instead of those 3, you might have 300. And so this is one of those ways in which that data file size can really add up quickly. There are several ways that hyperspectral data like this can be captured. So in this image, we've got this like push broom technique where the image goes in a line for capture. Filter based takes the whole thing at once, and a whisk broom kind of goes across the area that you're capturing the image of one pixel at a time. There's also non-imaging based data capture, so point measurements. Then you're getting basically like 1 pixel's worth of data from the image, and this might be averaged over an area that's going to vary depending on your spectrometer. But image-based spectral data capture gives you that extra dimension, that spatial dimension to your data set. But these sensors are typically more expensive than your non-imaging spectrometers. And this is not a hard and fast rule, it's just in general. but they may also require additional considerations for lighting and may not come with a light source integrated into the system. So that's something that you'll have to consider when working on your experimental setup. Point-based or non-imaging spectrometers can be, there's lots of different modes. So you can have handheld sensors, the more traditional benchtop sensors, or they can they can be mounted in line on a combine or on a processing line. And so they're pretty versatile that way. Typically less expensive, but that's not necessarily true. There are also very expensive versions of all three of these different types of non-imaging spectrometers. But often these do contain a light source, not always, but often. The next thing you'll need to consider is the number of spectral bands or wavelengths that your spectrometer is able to capture. And so there are two main categories for this, multispectral and hyperspectral. And I think it's really important to understand the difference between the two. So multispectral typically focuses on just 4 to 7 bands. So for example, this sensor from MicaSense has it captures light in blue, green, red edge, and near infrared, but it doesn't span the spectrum in between those points. And so this is a limited amount of information you're getting back that will probably not allow you to do your typical near infrared analysis methods. But this is very useful for calculating vegetation indices. And I'll talk a little bit more about vegetation indices in a minute, but it's a very established technology and method. Hyperspectral, on the other hand, has many more bands, up to hundreds or thousands. And this can be image or point-based. So as I discussed in the last slide, you can get just that one pixel or a whole grid of them. And this is useful for direct or traditional NIR prediction and also for phenomic prediction. When capturing your data, you also have to consider scale. So this could be landscape, field, plot, plant, leaf, sub-tissue level, all the way through. And the sensor that you choose is going to dictate what level of data that you can capture with it. So this can range anywhere from satellite data to handheld or inline sensors and everywhere in between. So these sensors could be mounted on a plane or UAV. You could set up a photo booth in the lab for them, or use your lab benchtop machine. There's a ton of different options, but this is going to dictate what then you can look at in terms of your analysis or your scale. So that's another important consideration. And lastly, with data capture, experimental setup is important. So temperature can affect the spectral reflections that you're getting back, and ambient light conditions as well, especially if your spectrometer doesn't have like a closed chamber that you put the sample in. You can prepare your samples in different ways. Some spectrometers will only work with like dried ground samples. And so this is where that non-destructive aspect of the spectroscopy breaks down. But a lot of them work with whole samples or can even be taken out to the field or into the greenhouse for data collection. So again, the type of spectrometer that you choose is going to impact how you need to be preparing your samples for scanning. And then also the number of samples that you're working with is important too. Some take more time for data collection, and so you need to be considering your throughput with that. There are several other practical considerations that may not necessarily affect the biological or physical data collection, but are definitely going to affect your budget or your ability to get through samples as a breeding program. So different spectrometers, like I said, can take different amounts of time for scanning, and so can affect your throughput when you're working on a batch of samples. They have different levels of portability and much different price points as well. So these, even just in this picture, range from about $1,000 to over 100,000. So there are lots of different options for many different budget levels as well. They all output different file types and formats, which can make analysis really tricky. And that's something you definitely want to consider, because some of the data types that are output are proprietary and require a translation step before you're able to use them with different types of software. So it's just something that you want to make sure that you're aware of before you actually start collecting your data. And you want to have a plan for how you're going to collect it in terms of software as well. So data collection software for spectroscopy is often included with the instrument, especially if you're buying a lab quality instrument. But they vary widely in terms of how easy they are to use and the format that the data is output in. And something I want to make sure that you note is that some spectrometers limit the access of raw data for the user, and so then you're stuck within that software for your analysis as well. And that's definitely something that you want to make sure that you're aware of before you start collecting your spectra. We've also found some spectrometers don't allow you to input your own sample names. They automatically assign a sample name like 1, 2, 3 to the samples as they're being scanned. And that can make things really difficult for data management later on, where you might need to record the sample name that the spectrometer is assigning, as well as the name of the sample, and match them up later. And while this doesn't make it impossible to do, it just it adds that extra step, which can really slow things down, and it adds another opportunity for mistakes to be made. There are several, or at least 2 limited open source options for spectral data collection, and I'll talk about Prospector on the next slide, and then Spectrograph later as well. But these are both free open source software. Well, I'm not actually sure if Spectrograph's open source, but it is free. But they're available on only specific platforms, and they only work with specific spectrometers as well. So Prospector is an Android app. It's free and open source and part of the PhenoApps effort. So Trevor Reif, who talked a couple of months ago, was the main point person on this, and I was involved a little bit in the app development, definitely in the testing. But this is an Android app for capture of spectral data. It doesn't do any analysis, just capture. And right now it only works with 2 different spectrometers pictured here below. One we found works a lot better than the other, so I would not recommend the link square on the left, but this InnoSpectra on the right does work pretty well, and I can give you more information about that later as well. The app, yeah, free on Google Play, and you're welcome to download and play around with it. But in order to use it, you do have to purchase a spectrometer, so don't expect it to just collect spectral data with your phone camera or something. Okay, so that was a lot to digest. There are just so many things to consider with data capture. Processing is a little bit more straightforward, but again, there are lots of different options and things to sort through. So I'll go through this next. I'm gonna skip this instrument standardization. This is like internal calibration of the instrument. So most have like a some kind of white square that you might scan before you start, or a big tarp that you put out in the field for correcting images. But this, it really varies from instrument to instrument, so I'm not going to address that too much. Okay, so image processing starts with the steps that you might follow for normal RGB image processing. And Heather talked about this a lot last month, but For things like UAV images, you're going to have to stitch them together and then parse out where your plots are within that image. And if you're taking images on a normal camera on the ground, you're still going to have to parse out the background versus your sample. And then from there, you can either aggregate your spectral data within your area of interest, so within your plot or within a specific tissue on your sample. You can make the decision of how you want to do that and then extract that data. And then you're at about the same point you would be if you were taking non-image spectral data, so those like point measurements that I've been talking about. Once you've got your spectra extracted and you've got one spectrum per sample unit, you can go through the outlier removal process. And actually, I typically do this before I aggregate my spectra, just in case there are any wild outliers that are going to affect then the mean spectra that I end up with. So if you look at this figure on the right here, there's this red line amongst the blue that's clearly very different from the most of the spectra that were captured in this data set. And there are several, well, more than several different methods you can use for outlier removal, but the most popular in traditional near-infrared spectroscopy analysis seems to be Mahalanobis distance. And this is a measure of the similarity or dissimilarity of a sample or an individual from a distribution. And it can be tricky to decide where that cutoff is between outlier and non-outlier. There are lots of different guides for how to do that if you look through the literature. So you're going to have to figure out for your use case and your spectrometer what works best for you. Unfortunately, there's not a super hard and fast rule for that. There are lots of different variations on Mahalanobis distance and many other ways to calculate your whether a sample isn't or a scan is an outlier or not. What I would not recommend, however, is just relying on visual inspection. So in this case, we can clearly tell that there's one extreme outlier, but in general, it may be easy sometimes to pick these out, but there might be some like fuzzy ones where you're not really sure if it's an outlier or not, and it could have some biological signal that's going to be really important for your analysis. So I would recommend not visually picking out outliers, but instead using one of the many methods out there for identifying the outliers in a more repeatable and less subjective way. Once you've got your samples all outlier free and looking good, you can then graduate on to the pre-treatment step. And with spectroscopy in general, this is a very common step that different groups go through. So pre-processing is another name for pre-treatments, but ultimately this step aims to increase the signal to noise ratio of your data set. So there are lots of different variables and things that can affect the spectra that come back to your sensor other than the biological signal that you're trying to pick up. These can include scattering from light. There can be baseline shifts that are from different particle sizes. You can just have weird artifacts from your spectrometer. And so these pre-treatments help, in theory, to reduce that noise and allow your biological signal to shine through. So in my experience, the best pre-treatment to apply really varies depending on what spectrometer you're using. And at times it can vary depending on the samples that you're scanning. So maybe if you're scanning leaf tissue versus root tissue, or if you're taking images from a UAV, like you might have different pre-treatments required. So Some of these include just taking the first derivative of your spectra or the second derivative. There are different algorithms like Savitsky-Golay or lots of different combinations of different types of pretreatments. So I've shown some of these on the right here, taking this raw spectrum and transforming it into these different pretreated spectrums. And you can see there's a lot of variation in what those look like. And some will end up working better than others for, for your use case. Unfortunately, it can be difficult to decide which one to go with as well. It's not something that you can just look at visually and be like, oh yes, this is the one for me. You really need to test it and see how much different pretreatments affect your accuracy of prediction. And that seems to be the best way to decide. Your spectrometer manufacturer in their proprietary software may have already figured out the best pretreatment for that machine. And so they might have already done that step for you. But if you're doing everything yourself, you'll definitely need to at least consider this step. So once you've got your data captured, pre-processed, you're actually then finally ready to do some analysis. There are, I'd say, 3 main types of analysis that you can do with near-infrared spectra. There are, of course, many other ways that you can visualize and play with the data, but 3 main ways that this might be useful for plant breeding. And so I'm going to just walk through those today. The first is vegetation indices. These, when I mentioned multispectral cameras earlier, this is a great use for those. So there are tens, if not, I don't know, maybe even hundreds of vegetation indices available. These are often just ratios of different spectral wavelengths with each other. So they don't require any prediction at all. You don't need to run a model or anything like that. Once you've got your spectra, you can just calculate it directly from the spectra. And these are often used then downstream as covariates and mixed models and things like there are lots of uses for vegetation indices, but I just want to make sure that the distinction is clear between vegetation indices and then the other methods that I will discuss in a second about for spectral analysis. So one main example of a vegetation index that you're probably all familiar with is NDVI, or normalized difference vegetation index. And this is the ratio of the NIR band of a multispectral camera minus the red over NIR plus the red. And it can be a measure of the greenness of leaves, which there might give you information about how healthy the plants are. So as opposed to vegetation indices, traditional near-infrared spectroscopy analysis involves the whole spectrum. So this can be done more for predicting quality traits of the sample that you're scanning, but it can only really tell you about the properties of the sample itself. So if you're scanning root tissue, it's not necessarily going to tell you about the chemical composition of the leaves unless you have some trait in your roots that is highly correlated to the composition of the leaves. And so if you're interested in understanding leaf quality, you're going to have to scan the leaves, not just the roots. And so I think this is important to note because you're going to have to collect a different data set for each tissue you're interested in looking at. Now, but you don't necessarily need to take new scans to look at different traits within the same tissue. Traditional near-infrared spectroscopy, spectral data analysis does require that model calibration. So you're going to have to collect some wet lab data or use whatever traditional method that you've been using to have a data set that you can use for training a prediction model. There are two main types of this analysis. You can use it for regression, so looking at a numeric trait like the dry matter content of a root or a leaf. And then you can also look at discrimination. So you could use it for telling the difference between two different types of soybeans or things like that, where you've got categorical variables. Traditionally, this workflow for this analysis goes from calibration. So you collect the data set that you're going to train the model on. This includes those reference samples using your traditional phenotyping method. And then you actually train the model itself, and you can do cross-validation within the training set or on a validation set. And then, and only then, can you use it for routine use in your breeding program. And there's a lot of protocol optimization that can happen between these steps. You may need to go back and collect more samples or collect them in a different way to try to improve your prediction model. But once that's set, yeah, then you can move on to routine use. But even then, you're not really set forever. I would recommend regularly updating your models, especially in a breeding program where your population is going to change over time. And hopefully you're moving it towards that, improving those quality traits, but then the range of values you're getting for a specific trait may change. Like maybe the protein in your population is going up on a whole. You want to make sure that your training set is representative of all of the samples that you're going to predict, or it's not going to do a very good job of predicting. There's a lot more that I could say about this type of analysis, but there's also a lot of literature out there on using this type of analysis and guides that you can find. There are lots of different metrics for deciding how well your prediction model is working. And unfortunately today, I don't have time to go through the mechanics of this, but feel free to ask me questions afterwards about it. So the third type of data analysis I'll talk on today related to spectral data is phenomic prediction. This is a pretty new method that's been used only in the past 5 years or so, at least published in the last 5 years or so. So this method is very different from the previous method we talked about. So the previous method, when you are creating a model, you're predicting traits on the sample that you scanned. So you're predicting root traits if you scan a root, or seed traits if you scanned a seed. But phenomic prediction allows you to scan one tissue and predict traits that might affect the whole plant or be in a different tissue or in a different environment. And it does this through using a relationship matrix. And this is familiar through, it's the same method that you might use in genomic prediction, but just a different way of calculating that relationship matrix that's used for prediction. So yeah, I don't know if I could go into a lot more depth here as well, but for now I will leave it at that and just say this is worth maybe looking into. It's not clear yet whether, it's also not going to be a panacea, and unlike there are a lot of potential problems with it as well. So just something to be aware of moving forward. So there's lots of options for analyzing your data in terms of software. There are a couple of free options and then some proprietary options as well. So I mentioned Spectroglyph when talking about data collection software. This is a free software download for Windows only, but it handles many different formats of spectral data. So even if you're not using it for analysis, it is really helpful for being able to translate between different file types. So between like a proprietary file type and a CSV, say. But it has some analysis capabilities. They are limited. And maybe not quite as flexible as programming an analysis pipeline for yourself, but it is really great for quick visualizations and maybe even for some preliminary analyses. My go-to is R. You can also do any of these types of analyses in Python as well. I mean, these are, super flexible programming languages, and if you do it the right way, at least you're going to get something that's really repeatable. And so if you need to tweak a parameter in your pipeline, it's really easy to just run it all again. And that to me is the biggest plus to using a programming language or a script rather than using a GUI or graphical user interface software. There are lots of different packages available, especially in R, for spectral data analysis. And I'll talk about one of those, but there are lots that are helpful for different points along the way. So some packages might help with your pre-treatments, some with your model development. And then during grad school, I put together this wrapper package that puts together several of those different functions into a pipeline that makes it fairly easy. Of course, I'm biased, but easy to plug in your data set and get some predictions out to know how well your spectral data is doing at predicting your trait of interest. It also has the capability to save the prediction model once you're happy with it so that you can use it then for predicting and you don't have to go through the training step every time you want to use your model. So this is free on CRAN if you're interested. There are other options of analysis software that are not free and open source. Often spectrometers will come with analysis software. They vary in terms of how easy they are to use and how flexible they are, how many different parameters you can change. So they might be easier to just get up and go and use it. Less setup, definitely, but they may be less flexible and may not let you do all the different types of analysis or compare different parameters the same way that you can with R. Many companies also offer model development as a service. So you could either send your samples off to them and they will do the scanning for you, or you can scan them in-house and then send the data off to the companies to do the model development. And that can be really useful if you, instead of wanting to focus your time on learning all of the different aspects of analysis, would rather just spend some money and get it done well. And I don't have any recommendations on specific companies to reach out to for this, but it is available. And just do your own research on that and see what you can find. So finally, we'll get into data storage, so file transfer, formatting, and long-term storage. I've mentioned this a couple times now, but different spectrometers often have their own file types, which makes integrating them a lot of fun. It really doesn't. It can really make things difficult if you're trying to compare different spectrometers or use any open source software. So I mentioned Spectrograph can help with translating between these different file types. I would highly recommend that. There may be other methods as well, but that's worked really well for me. And then if you're interested in using an open source platform or R or Python, CSV is the handiest to use. And so, you'll have to convert the data type to CSV to be able to use them. There are a few packages in R that can read in some of these data types, but it can get pretty messy. So I would recommend using Spectrograph. But typically your spectral data is going to be laid out in a format similar to what I have here at the bottom, where you've got each row is a sample. And then you may have some metadata on the side, like a timestamp, your sample ID, maybe what tissue you scanned. You could put your reference data here as well. and then spectra going to the right. And so you'll end up having one column per either wavelength or half a wavelength, whatever unit your spectrometer is measuring. Some have them flipped, or where you've got your spectra on each row and your samples on each column, but you'll just want to be aware when you're looking at spectral data which way it's oriented before you start. So spectral data files can be large, and especially with hyperspectral images where you've got that full spectrum for every pixel in every image. So you'll also just want to come up with a plan before you start of how and where you're going to put your data. Where do you want to keep them long term? Just make sure that you have enough storage space, and that you budget for storing it if you're going to need to purchase more hard drive space. A lot of spectrometers come with their own databases for storage. This is especially true of benchtop spectrometers, but the handheld ones often do not. So you'll want to just, again, come up with an organized plan, a way to name your files that you're going to be able to find the samples that you want later on. Ideally, we would be able to move around this data with Brappy to different devices and databases. This is under development. We've got something that's working, but it's not officially part of the breeding API spec yet. So be looking out for that. Hopefully it'll be coming soon. We have, however, integrated spectral data storage into BreedBase. So this is a figure from a manuscript that describes the spectral data storage and analysis in BreedBase, but basically you can upload your CSV files of your spectra into BreedBase, and then there are some limited options for spectral data analysis that use my R package in the back end to make predictions that can be saved and then used as you would normal phenotypes in the database. And this is what the interface looks like for the analysis, where you can choose your parameters and make your predictions. It's fairly straightforward, but I will say it still has a few bugs that need to get worked out. So I don't necessarily recommend relying on this as the sole source of your data. You should definitely keep all of your files if you're using this software. Okay, quick before I'm done, I'm gonna talk through a couple of case studies of ways that I've used spectroscopy in my work, so that you might be able to get a picture of how this might apply to your breeding programs. First, I used it working with cassava. So cassava is a staple crop. It's a starchy root that grows like a shrub. And some of the primary breeding targets for this crop: pest and disease resistance, yield, performance, and dry matter content. So the opposite of the water content of the roots. Dry matter content is a major part of both the dry yield, which is used for making flour, things like that, and also the food quality. So cassavas can be eaten, cassava root can be eaten like steamed, and it affects the mouthfeel of the root. But unfortunately, this trait can be time-consuming and laborious to measure when you're looking at a breeding program scale data collection. So these photos are from the National Cassava Breeding Program in Uganda during a visit that I took. So roots are hand shredded, they're dried in big drying ovens, and they're weighed before and after drying. And the dry matter content is the ratio of the two. So we tested the use of handheld spectrometers, the SIOs, to predict the dry matter content of sliced cassava roots. This took only about 2 minutes per sample or less, and we made prediction models using the spectra to predict that oven dry matter content using the traditional shred and dry method. I tested a partially squares regression model to predict the spectra, and then we looked at it and different field trials. So this is data from IITA in Nigeria, but over 10 different field trials, we got pretty good results for our predictions of dry matter content. So this was the first time testing mobile spectrometers of at least commercial grade, or yeah, like handheld consumer grade handheld spectrometers for predicting this trait in cassava, and it looks pretty promising. So this may not be, you know, you're not going to get the same accuracies as actually using the oven method, but it could be used for a level of culling or a selection in the program. So I've recommended to the group at IITA that, we do routine model updates. There seems to be some variation between different field trials and environments. And as I said, their populations are changing as they're making selections. And so making sure that we are really representing the full range of those reference values in our training set is really important. Second case study I'll look at is using phenomic selection at the Land Institute. And so this is what I was working on during my postdoc. So the Land Institute is a nonprofit in Kansas that's working on domesticating perennial grain, oilseed, and legume species for use as crops. And one of their main bottlenecks right now is that only one of these species that they're looking at has a reference genome, and so a tool like genomic selection is not available to them yet. They're working on it, but they're not quite there. So we're comparing different methods for spectroscopy-based phenomic selection in this project using the high-tech LEM-detect system at the Danforth Center, and then also some of these handheld spectrometers that I've mentioned. So we did a pilot study with Kira clover, which is a perennial cover crop with limited genomic resources. So we had clover plants in the greenhouse. We had more than 200, but this was a subset of them that we were specifically looking at, and we took 3 different phenotypes here with varying levels of heritability. We scanned them with 3 different types of mobile spectrometers, or well, 3 different spectrometers, 2 are mobile. So these are the 3 spectrometers that we were comparing. We have the SIO, which is the same one that I used for the cassava study, the InnoSpectra, which is one of the 2 that works with the free Prospector app developed by the PhenoApps group, and then the FOSS DS3, which is more lab grade benchtop spectrometer that is considered by many to be state of the art, but does require the samples to be ground or homogenized, at least when scanning leaf tissue. So looking at the results here, this is a subset of our data that we are working on expanding. We see that the accuracy of the method and of the spectrometers really varies across the traits, across the different spectrometers. So we saw that the FOSS instrument was great for biomass compared to the other 2, but the much more inexpensive spectrometers were keeping up with it for the other traits. So lots of exploration left to do, but this will just give you a little taste of the phenomic selection. So final thoughts. Make sure you plan out your experiments before you start. Choose the right spectrometer for your use case, and just think through all the different considerations that I've mentioned today. It's worth going through a test run with a small batch of samples to make sure that things are working well for you before you decide to scale up. And I'd recommend looking through the literature to see if near-infrared spectroscopy has successfully been able to predict the trait that you're interested in before making any purchases, if possible. routinely update your models if you're using the traditional near-infrared spectroscopy methods. And yeah, you're going to have to validate each new spectrometer that you're looking at with every trait and crop combination. It's unfortunate that it requires that much work, but it does. Lots and lots of people and funding sources to thank, but I am just about out of time, so I'm going to leave it at that. And